familiar with sudden oak death. Sudden oak death is a relatively new forest disease caused by the introduced pathogen Phytophthora ramorum. Since the mid-1990s, when it was first observed causing mortality around the San Francisco Bay Area in California, this disease has killed over 1 million tan oak and true oaks in California's forests. It's still not known where this pathogen was introduced from. It's definitely not a native pathogen. It was introduced to California on nursery stock on infected plants, but the origin of this pathogen is not yet known. On the map on the right, you can see where the current distribution of Phytophthora ramorum and in turn sudden oak death is in California. Definitely centered around the San Francisco Bay, down in the Santa Cruz Mountains, down in Big Sur. I'm moving the arrow down there. Maybe you can't see that. Anyhow, and then working its way up Sonoma, Mendocino, and into the north coast, including in the southwestern portion of Oregon. So this disease, when we get on the ground and see what it's doing, it is a really huge disturbance to California's coastal forests. It's really wreaking havoc in redwood tan oak forests as well as mixed evergreen forests. You can see from this series of photos that it kind of goes, the mortality goes through progressions. It starts with the trees die standing in place with all of their leaves and the fine fuels, and as they gradually decay and fall down, then we get more ground fuels, and really does quite a number to the forest. So a little bit about this pathogen. Phytophthora ramorum is an interesting pathogen. It's fungal-like, but it's not a true fungus. It's considered an oomycete pathogen. Like fungus, it uses spores to reproduce and to spread. In this photo right here, that is a sporangium with zoospores coming out of it. Zoospores are water-adapted spores. They actually have flagella on them, and so they can actually kind of swim in water, and so this pathogen is really adapted for wet environments. These spores are formed in wet environments, and Phytophthora ramorum is mainly aerially spread during rain events and wind. So some of the hosts that are important in epidemiology of this pathogen, coast live oak. This pathogen kills coast live oak. It causes these bleeding stem cankers, which eventually girdle the tree and kill it. And it's important to note that this host, for example, is a dead-end host. The pathogen can't sporulate from it. So the pathogen kills these trees, but it's not using these trees to then reproduce and spread to other trees. In contrast, the California bay laurel is a sporulating host only. This pathogen can sporulate abundantly from leaves of California bay laurel, but it does not have any adverse effect on the fitness of this tree. Then we have tan oak, which is in a way the most susceptible host to Phytophthora ramorum. It's killed quite easily and readily by the pathogen with the same mechanism in coast live oak. The pathogen infects the stem of the tree, causes these bleeding cankers that can girdle the tree and kill it. However, the tan oak is also a sporulating host for ramorum, so ramorum can infect the shoots and the stems and the leaves of tan oak and then sporulate from there and spread to new hosts. Then there are a lot of other hosts that are kind of minor hosts, but that do play a role. For example, canyon live oak, twig dieback can be caused by ramorum. And then there are a whole bunch of other hosts that are also minor hosts that symptoms are not always seen, not so pronounced, but the pathogen can infect these. Douglas fir is one of them. And there's a wide number of native hosts in California that can support this pathogen. It's also important to note that this pathogen is not just in the west coast of the United States. Right now, it's in England as well, where it's causing mass diebacks of Japanese larch in plantations. So this pathogen has a very wide host range and is something to watch in the future. So our study area is the Big Sur region, located on the central coast of California in Monterey County. It's an incredible landscape of really great biological diversity, a really wide variety of plant communities and habitats. It's been recognized as a hotspot of rarity and richness by the Nature Conservancy in an area that conservation should be a priority in. Yet the ecology of this region is pretty poorly studied, and unfortunately, this area has been infested early on with Phytophthora ramorum. So it was one of the areas in California that probably had direct introduction of the pathogen on nursery stock, probably sometime in the 1990s, and the pathogen has taken off from there, and there has caused very high levels of tree mortality in some stands. So because of these reasons, a plot network was established in Big Sur in 2006 and 2007 to look at the impacts of sudden oak death on the landscape. Plot network consists of 280 plots. These plots are stratified over watersheds, forest type, fire history, and areas of sudden oak death mortality. So as you look on this map, you can see where the distribution of all those little dots are plots in the Big Sur area. A lot of them were known to have P. ramorum in them at the time of establishment, and a lot of them were considered free of disease at the time of establishment. In terms of forest type, so I mentioned that redwood tan oak forests as well as mixed evergreen forests are the main forest habitats where P. ramorum is found. So in the Big Sur area, redwood tan oak forests tend to be down in riparian zones and in deep valleys or closer to the ocean, whereas the mixed evergreen forests tend to be up more on slopes and a little higher and drier than the redwood tan oak forest. But both of these habitat types harbor the pathogen and have seen very high levels of mortality in some areas. So a bunch of baseline data was collected at the time of plot establishment. All stems greater than 1 centimeter DBH were measured. The P. ramorum symptoms and incidence of disease in individual trees was noted within each plot. So we had a lot of information at the time of this, that this network was established. And then in 2008, really just a year or two after the plots were set up, wildfires really took a hold in California in that summer. There was an early start to the fire season in 2008 following 2 years of really extreme drought in California. On June 20th of 2008, there were dry thunderstorms throughout the state, which the lightning strikes from ignited a lot of different fires, over 2,000 fires. And in this map, you can see all those red dots indicate where there were fires burning a few days following those storms. So we're going to zoom in down in the Big Sur region where there were a few fires going on. One of them, the Indians fire, actually started prior to these storms. That started in the beginning of June. But then when the basin complex fire was sparked in the June 20th storms, it eventually merged with the Indian fire. And the two combined to a kind of a super fire. They weren't contained until July 27th, and the combined fires ultimately burned 244,000 acres, which makes it one of the largest recorded wildfires in California's history. About $120 million was spent on suppression of this fire, which makes it, by some estimates, the most expensive wildfire in California's history. So how did these fires impact Big Sur and our plot network? Well, first off, they were the first major wildfires to occur in forests infested with Phytophthora ramorum. The basin complex fire alone burned about 163,000 acres. And then there was actually a smaller fire that started in September to the south of the basin complex perimeter, which was called the Chalk Fire, and that burned an additional 16,000 acres. In all, 121 of the 280 plots in the Big Sur plot network were located inside of the fire perimeters. So all in all, this worked out to be a really unique research opportunity. And this is kind of when I arrived in California to this project. Before that, I was on the East Coast and thought I was coming out to California to do a master's degree on cenotes up in the north coast of California. But I arrived and I was told, hey, we've got a really unique research opportunity. Fires have swept through our plot network in Big Sur. We have all this pre-fire data, and we want to learn something about it. So just to give you a little background of how I stepped into this project. So seizing on this research opportunity, Rizzo Lab, pretty much right after the fires were contained, so starting in September of 2008, a lot of people went down there and we were taking burn severity surveys in the plot network. In some cases, you can see from that photo on the upper left-hand corner, we were going into areas where fires were still smoldering. And in the months of September and October, before the rain started, we were able to survey 61 plots that were burned in the basin fire. And we took standard burn severity measurements such as ash depth and tree scorch heights. We rated fire damage to 5 forest strata using continuous variables. And then we kind of combined the fire damage ratings from these 5 strata, the substrate, herbs, shrubs, intermediate trees, and dominant trees, to calculate a composite burn index that could give us an overall assessment of burn severity damage in a plot. So what we had essentially was a huge amount of knowledge of what was going on in these areas before the fire, as well as a lot of information about the impacts or the burn severities to these plots after the fire. And we could take this information in a lot of directions. Some people wanted to know how did the disease impact fire severity? Did the fires burn hotter and more severely in areas where there was the disease and the disease had caused a lot of fuels to build up? I, however, being more of a plant pathologist, took the kind of flip side to that question and wanted to know, first off, what was the overall impact of the wildfires on Phytophthora ramorum survival? Was this pathogen able to survive in areas that were known to be infested prior to the fires? And if the wildfires did not eradicate the pathogen, under what conditions was the pathogen able to persist in forest stands? And what are the likely reservoirs for pathogen persistence and reinvasion following fire? So my methods to accomplish these objectives were, I chose 63 plots in our Big Sur network that were all known to be infested with the pathogen prior to the fires, and I surveyed them both 1 and 2 years post-fire. So there was exactly 45 burned plots and 18 unburned plots, and these plots were equally divided between mixed evergreen and redwood tan oak type forests. In 2009, we sampled all vegetation that had symptoms of Phytophthora ramorum in all of the plots. And so in terms of how we sample and detect Phytophthora ramorum, we take vegetation. Here you can see that here's a bay tree with lots of ample sprouts following the fire. So if any of these leaves had symptoms of the pathogen, we would sample those. We bring them back to the lab. We cut up little pieces. We insert them into a selective media that only allows Phytophthora species to grow in it. It doesn't allow other fungi or bacteria. And so then we use microscopic identification of the pathogen growing out of those tissue sections to determine if indeed it is Phytophthora ramorum. So we did that kind of sampling the first year as well as the second year following fires. In the second year following fires, we really focused on those plots from which we hadn't been able to recover the pathogen in 2009. And we also resampled 11 control plots in 2010 that had been positive in 2009, just to make sure that if a plot was found to be positive for the pathogen in 2009, that it was indeed staying positive the following year. So the overall story from our recovery results was that P. ramorum was recovered even in the burned plots, but recovery in burned plots was disproportionately low compared to unburned plots. So in 2009, we recovered the pathogen from 20% of our burned plots versus 72% of our unburned plots. That's a much smaller recovery than one would expect by chance. And in 2010, the pathogen recovery doubled in burned plots to up to 40%, but it was still a lower recovery than could be expected by chance compared to the unburned plots. If we looked at how many trees were actually from how many trees did we detect the pathogen in burned versus unburned plots, there was actually, if you just looked at those plots from which we did recover the pathogen, there wasn't really a difference in recovery between burned and unburned plots. So here on the left, you can see in 2009, 1 year post-fire, you see burned plots. In some of these burned plots, the black bars indicate that we were getting nearly 100% or at least over 50% recovery of the pathogen from different plants that we sampled, and it really wasn't very different than what we were seeing in the unburned plots. The same trend was evident in 2010, but you can see that we were able to recover the pathogen generally with a higher frequency from all plots, unburned and burned. So then to answer the second question of my research objectives, so we knew that the pathogen had in fact survived on the landscape in certain areas, but we wanted to know, was there something different about the areas where we were able to recover the pathogen post-fire as compared to those areas where we couldn't recover the pathogen? So because we had so much data from before the fires and after the fires, we kind of took out a bunch of plot level variables that we thought would be possibly influencing pathogen survival. And I've kind of split them up into a few different categories. So we had different burn severity indices, based on those burn severity measurements that we did immediately post-fire, as well as looking at dead basal area of P. remorum hosts. So that was looking at cumulative mortality between 2006 and 2009 of what portion of the basal area of those hosts had died, presumably because of the fire. We also looked at pre-fire host density variables of both bay laurel and tan oak, post-fire host density measurements. We looked at pre-fire disease levels, so how many trees in a plot demonstrated symptoms of the disease prior to the fire, and we looked at landscape factors. So these variables here that are circled in red are the variables that we ultimately ended up identifying as what we thought, what we hypothesized would be important on disease recovery. We chose these factors based on what we know about the biology of the pathogen, as well as doing some just very basic statistics, t-tests, looking at how these measurements varied between burned and unburned plots. And we took these variables and we put them into a multiple logistic regression model. So that is to say, we took variables that we thought would be important on influencing pathogen recovery, and we put them together in a model, and then the outcome of the model is going to help us to determine which of those variables had a significant impact on recovery of the pathogen, which of them were good predictors to decide if the pathogen was going to survive or not. So what we found in 2009, burn status, whether a plot was burned or unburned, was very significant in determining whether the pathogen was there, not surprising. And we also found that of all these other variables that we put in there, the number of symptomatic bay laurels that were in the plot prior to the fire was also a very significant predictor variable. So what the odds ratio can tell us, and the odds ratio is calculated on output parameters from these logistic regression models, they can kind of help us gauge what the impact of those factors were on pathogen recovery. So in the case of burn status, when you have an odds ratio of 29.117, what that's saying essentially is there was the chances were 29 times greater of recovering the pathogen in burned plots compared to unburned plots. In unburned plots compared to burned plots, excuse me. And with an odds ratio of 1.237 for symptomatic bays, that's essentially saying with increasing numbers of bay, you're going to have an increasing chance of recovering the pathogen post-fire. In 2010, when these same predictor variables were put in the model, those that came out to be significant predictor variables in the model were forest type this time, as well as the proportion of dead basal area of bay laurels. So once again, the odds ratio here tell us for forest type that is roughly 11 times more likely to recover the pathogen in redwood tan oak plots versus mixed evergreen plots. And the odds ratio for the proportion of dead bay basal area, the fact that that odds ratio is less than 1 tells us that you had an increasing likelihood of recovering the pathogen when you had lower levels of dead bay basal area. So to help kind of visualize that rather dense chunk of data, we can graph the output of that logistic regression model. And what we can see here in the panel A is that this is in 2009 in response to varying numbers of pre-fire symptomatic bay laurels. What you see is that the probability of P. remorum presence in a plot increases with increasing numbers of symptomatic bays. And that furthermore, so the solid red and the solid blue line, which have the highest levels of probability at any point, those are in unburned plots. So it's showing that the probability of pathogen recovery in unburned plots is always higher than those in the burned plots. Then when you look in panel B, in 2010, if we're looking at the probability of P. remorum presence in response to varying proportions of dead bay laurel basal area, you see that you have in the beginning your highest probability of detecting the pathogen when you have very low levels of dead bay basal area, and as those the amount of dead bay basal area increases, your chances of pathogen present drop off. And that here, the two top lines, the solid red and the dashed red, those indicate that those are in redwood tan oak forests. So here it's showing that there's always that increased probability of pathogen presence in that forest type as compared to the mixed evergreen forest type. So on to another dense and complicated figure. This is, we did NMDS ordination, which is essentially you take all of your data points, so each one of those little symbols on the graph represents a plot, and we used all of the 24 different plot level variables to scatter our plots on this graph, and then you can overlay the different variables on them and group them and do a lot of interpretive looking at how things are scattered. And I'm just going to tell you the overall take-home message from this ordination is that pathogen recovery 1 year post-fire is in plots that had the lowest burn severities. Pathogen recovery 2 years post-fires, that is to say, in those plots where the pathogen could only be detected, not in the first year, but only in the second year post-fire, those plots had greater burn severities, and pathogen recovery was largely influenced by high levels of that pre-fire disease prevalence and low levels of post-fire bay laurel mortality. In plots where there was no recovery of the pathogen 1 or 2 years post-fire, those plots had the highest levels of burn severity as well as post-fire bay laurel mortality. So on to my third research objective, which was trying to determine where exactly was the pathogen able to persist in these burned areas. We compared pathogen recovery in different types of tissue that we sampled. So I mentioned that when we were sampling tissue, I showed a photo of a bay that was sprouting, and I said, we could sample from those sprouts. Well, likewise, a lot of times we go into burn plots and there was still green foliage on the trees. Some of it was scorched, maybe half the tree would be scorched, but there would be some green stuff in there. So we could sample that, or we could sample newly sprouted vegetation that grew obviously after the fire. And we were very careful to note when we took samples where that vegetation was from, whether it was, post-fire regeneration growth, or whether it was likely, crown growth that had been on the tree probably at the time of the fire. It's important to remember that bays and tan oaks, these are evergreen trees. They don't lose their leaves, but they all are very prolific sprouters following fires. And what we found was that in burned plots, in both 2009 and 2010, we were able to recover the pathogen at twice the level from the bay laurel crown leaves compared to basal sprout leaves. But we found the opposite trend for this tree in unburned plots, as well as for tan oak and all plots. So let me just rephrase this in perhaps a clearer method, clearer way. Post-fire, when we go into these burn plots, we were able to recover Phytophthora morum from crown leaves on bay trees that had probably lived through the fire, but we were very infrequently able to detect it from this new growth sprouting from the base of the trees. However, with tan oaks, we most frequently detected the pathogen from the sprouts that had grown since the fire, but we weren't able to detect it very frequently from those twigs in the trees that had happened to live through the fire. And then something that we didn't expect to come across and weren't really planning on looking at, but we ended up finding, especially in 2010 when I really started paying attention to these other species, that we were recovering a few other Phytophthora species from this vegetation. Interestingly enough, none of the plots that were used in this study were known to contain either of these pathogens prior to the fire. So these pathogens, Phytophthora pseudosyringae and Phytophthora nemorosa, are also believed to be invasive or non-native Phytophthora species, but they certainly don't cause the mortality levels that Ramorum does. However, we seem to be, as we look for Ramorum, we seem to be finding them more and more within our California forests. So we started finding these other Phytophthora species, and what we were finding was that we were finding them overwhelmingly in the burned plots, and that we were recovering them predominantly from these bay laurel basal sprouts. So in that tissue type where I wasn't recovering Phytophthora ramorum, I was recovering these other Phytophthora species, which hints at some interesting maybe competition or interaction or something going on there that is unexpected and warrants further research. And in fact, I think that research is starting to take place right now. So in conclusion, we can say that the wildfires suppressed Phytophthora ramorum, but they did not eradicate it from the landscape in Big Sur, unfortunately. A lot of people had hopes that maybe wildfire was the panacea for this invasive forest disease, and if we allowed, wildfire to go to burn freely in these forests, that might help to take care of the problem, and doesn't seem to be the case. The pathogen was recovered both 1 and 2 years post-fire from vegetation growing in burned forests, and in some cases with no difference in frequency than from vegetation in unburned forests. Multiple interacting factors were responsible for the persistence or lack thereof of Phytophthora ramorum in these burned areas. Obviously, burn status and severity did have an impact, especially that very first year following fire. But forest type also played a really important role. The prevalence of hosts, inoculum pressure, that is to say how much disease was there in that area prior to the fire, and weather certainly plays a role. I didn't really talk about this, but there was a drought in 2006 and 2007, and for a pathogen that likes wet conditions, those conditions were not good for Phytophthora ramorum. So even in unburned areas, we weren't able to recover the pathogen that much. But once the rain started in 2009, pathogen recovery really went up in the forests around there in both unburned and burned areas. So obviously the start of more normal rain season was boosting the comeback of the pathogen as well. We noted that high levels of pre-fire bay laurel basal area in symptomatic individuals, symptomatic bay laurels, and low levels of bay mortality post-fire were important predictor variables in determining whether an area would whether the pathogen would be recovered in an area. and that the patchy burn patterns typical of mixed severity fires resulted in green islands, or refugia, where P. remorum could survive and spread from in these areas. So this is a photo of a burned redwood tan oak forest. You can see all the resprouting on some of those burned redwoods there, but where that big arrow is, you can also see that is a big green bay laurel tree that was just totally skipped over by the fire, whereas the rest of that area was burned pretty severely. And so we saw this a lot. You know, there was just a patchy burn pattern, and so if you have a big green bay laurel like that that just happened to be skipped over by the flames and is left on the landscape, and that tree was totally infected with the pathogen, that really serves as this refugium for the pathogen, and that as vegetation begins to resprout following the fire, the pathogen can just kind of use that as its focal point, and that bay is going to be sporulating, and the pathogen is going to spread from there and can easily reinfest the new vegetation. So here's another area, another photo showing one of our plots, and you can see it wasn't, I mean, this is kind of an edge of a burn, a perimeter. That looks like a very large kind of refugium in there, a lot of green plants, and certainly if that was close to any of our plots, that would be harboring the pathogen if it was previously infested, and spores with a wind event could be just carried from that green area into newly regenerating growth. So the sprouting of the Phytophthora host following fires, they provide a large quantity of tissues for Phytophthora to infect, though these other Phytophthora species may be competing with Phytophthora to infect some of these newly sprouted tissue in burned areas, and we don't really know what kind of competition is playing out in those tissues. So with that, I am going to acknowledge a lot of funding sources, USDA Forest Service Pacific Southwest Research Station, private and state forestry, an NSF grant, the Gordon and Betty Moore Foundation, and then I want to especially thank members of the Rizzo Lab who have been so helpful to me for doing all of this research, members of the Meitenmeyer Lab, which is a co-collaborator on the Big Sur Plot Network, and then California State Parks, Los Padres National Forest, Landals Hill Big Creek Reserve, Monterey Peninsula Regional Park District, and numerous private landowners in the Big Sur region who were so kind to allow us to do this research on their respective lands. And the research that I presented was just published, though, in this paper in New Phytologist, and there's the reference. And then the reference below there is the flip side of my research, looking at how sudden oak death impacted wildfire severity in the same plot network. So that's research by Margaret Metz, and you can look at that paper there if you want to hear the flip side of this interaction. The link suddenoakdeath.org is just a great comprehensive link about all things sudden oak death. And if anyone wants to get in touch with me for questions post the question session, or if they want PDFs of either of those two papers above, feel free to contact me at that email address and I can look you up. Okay, thanks.
All right, thanks, Maya. That was a great presentation. So I'll leave these citations up on the screen for a little bit before we switch to the next one so people can copy those down. And I'll also send those out in an email to everyone who registered for the webinar. So the first couple of questions, and I'll just pull these over where everyone can see them, relate to weather. And you may have addressed some of them, but the first one is, from Eileen Thiele. Was there any pre- and post-fire rainfall averages measured and recorded?
We did not use that kind of information in our analyses, and I mean, we could get it from different weather stations, but Big Sur is such, I mean, our plot network is spread out over such a big area down there, and Big Sur is so heterogeneous, and you got those little microclimates that I think it would be difficult to incorporate that kind of data across the board, but you know, I think it would provide some interesting information.
And then Joe Christie just notes that up in Bonnie Doon, where he is there, that there was 2010 was significantly wetter than 2009. And would that have affected any of the findings?
Yeah, probably. Yeah, I think I mentioned that when you have that increased rainfall, that we were getting greater recovery of the pathogen in both burned and unburned areas in 2010. So that kind of wet year, it confounds everything a little bit, and it makes it difficult to determine if it's just that the pathogen is infilling these burned areas because of time elapsed since the fire, and it's just taking that amount of time for the pathogen to reinfect new vegetation, or whether things were just so dry that the pathogen wasn't doing much even in unburned areas, and it really took that rainfall to kind of ramp up the pathogen's activity, and then we saw, you know, a big increase in symptoms, and were able to recover the pathogen more. So weather and rainfall is definitely a confounding factor in all of this.
And then just one final weather-related question from Ian Livingston. He writes, a related question to Eileen's, do you know if there's any correlation between foggy days as a relevant variable to sudden oak death?
Fog is certainly probably plays a role, but once again, this would be a little difficult because we had our plots were so scattered between, you know, lower and higher elevations, and in Big Sur, you know, you get the fog at the low elevations, or sometimes the opposite, and it burns off at different times of day. And so I think it probably affects the pathogen, but it would be really hard to incorporate that kind of data and that microsite data into these kinds of analyses.
Yeah, hard to capture quantitatively.
Yeah.
Okay, and then Joe Christie had a question about the meaning of the horizontal axis in the Phytophthora murmurum recovery histogram graphic.
Yeah, so they're just simply sorted by the number of samples that we took. So there's no order. It's only, you know, in those plots where we sampled the most because they had the most symptoms, those were at the front of the line, and then we just kind of ordered them sequentially with decreasing numbers of samples taken. So in some of those histograms, there's no bars because we didn't sample at all in those plots because either there were no symptoms to sample, or in the case of 2010, because we had declared the plot positive in 2009 and didn't resample some of those plots.
Okay. And Mark wrote that you answered this next question from his, so if I don't hear anything else from him, we'll just skip that. And good job on answering it ahead of time. And then Ian, we had that question already. And then Jake had a question. Are tan oaks expected to become a dead-end host?
No, in a sense, because as tan oaks in an area that's had the disease for a long time kind of all die, you know, they keep re-sprouting, and those sprouts can serve as sporulating. Well, you know, it's not really known. We don't know once the disease has really wiped out all the tan oaks in an area, how long they're going to continue to re-sprout and be able to serve as sporulators in that way. I think they'll definitely be able to do that for a while, but will that tree eventually become exhausted as its sprouts are repeatedly killed back and then it has to resprout again? But definitely for a while, those resprouts on the tan oaks, it was shown from this research that they become infected, very quickly after the fire, and so they could spread it to other low-lying vegetation, but because they're low-lying and not up in the canopy, they're not going to be as effective in dispersing the pathogen.
Yeah, we'll certainly find out further studies. And then Kent, a similar question, writes, why do you think the tan oak basal sprouts were more susceptible to Phytophthora murmurum?
Tan oak basal as compared to the bay laurel?
Also, or maybe the basal sprouts as compared to the crown leaves.
Well,
Kent, feel free to specify.
Yeah, so research has been done by Everett Hansen, who's a Phytophthora researcher up in Oregon, on the susceptibility of different vegetation types. And he found that young tissue is very susceptible to pathogen infection. So you have that young succulent tissue of those new resprouts, and you can imagine that that is something that's really, it's going to be easy for the pathogen to get into. It's low to the ground, so the pathogen can just kind of rain down on it from upper story canopies. So it doesn't surprise me that the tan oak sprouts are very susceptible to getting infected, but it makes the question of why aren't we seeing remorum in the bay laurel basal sprouts all the more interesting, because research from that same group up in Oregon also showed that likewise bay laurel basal sprouts and new growth is susceptible to infection, but we weren't seeing that. We were seeing, that we were getting these other Phytophthora species from the bays, but not remorum.
And that question is an excellent segue to this next question from Mary Huffman, which is, what research questions about sudden oak death are you most interested in? I'm sorry, what next research questions are you most interested in?
Ooh, that's a good question. I think there's a lot of unknowns. I think it's the big questions. I'm not so. We are looking at, in my current job, some of the more on the ground questions of, how can we prevent. For example, how important is movement of the pathogen in soil? That's something that hasn't really been figured out yet very well. We know you can move the pathogen in soil and that it's found in soil, but it's not really been shown whether that's an effective means for the pathogen to then initiate new infections in an area. But I guess the real big questions, and I don't know if this is something that one can do research on, or if it's just time will tell, is how is this going to play out? And as someone I think I noted in the chat, they said it's going to become another case of like chestnut blight. I think that will be the most interesting question is, what will this landscape look like in 50 years? And maybe I'll be around to see that.
Yeah. And Joe Christie has one more question. He said we can punt if we wish, but I'm going to go ahead and ask it anyways. It was a follow-up question on the weather. Curious about any significant climatic differences between the Santa Cruz Mountains and Big Sur. Just offhand, if you know if there's any significant difference in rainfall between the two areas.
I'm not even going to take a stab at that. I don't really know.
Okay, I can see Mark Reedy just chimed in saying you can use the data from the Big Creek Reserve, where
yes.
I believe he works. Yeah, thanks, Mark. And last question here, and feel free to type in any last questions in the chat box. While we do this one is from Kate and Eric Moore, and it is, would the changed tree spacing post-fire be a factor in changing the rate of infection?
Well, I think as the thing with these forests is that they all re-sprout all of these hosts. So it doesn't take very much time following post-fire before things get very dense once again. And I think when you have a kind of dense forest and things are close together, that it's easier for the pathogen to move around. And so certainly in the immediate post-fire landscape, where things are more open, drier, that is not as good an environment for the pathogen. But unfortunately, it doesn't take a lot of time before these areas kind of get dense again, and especially, I mean, because of the way that all of these trees resprout, and the fact that the fire was so variable and it really didn't uniformly, incinerate big areas. There were areas where some of the trees weren't burned at all, as I said, or they were just scorched enough so that they could actually resprout up in the canopy. And so I don't think that that impact of the fire will have very lasting effect on the pathogen.
Yeah, I would imagine it's another one of those things that's just really hard to tell.
Mm-hmm.
Yeah. And then we've got another question from Christine. These questions keep coming in. This is great. You can tell it's just a new area with a lot of work still to be done. And Christine writes, do you have any general guesses regarding the bay laurel sprout's lack of susceptibility, such as leaf compounds or structures, etc.?
I don't. It is my understanding that in Dave Rizzo's lab, he has a new student right now who's going to be following up on this, the interaction between the different Phytophthora species and what's going on. But in terms of the actual physical properties of bay laurel leaves, I don't know if there are any of those that might be impacting it. I'm guessing it's more of a competition between other, pathogens that are result in the lack of pyromorm infections that we were seeing, rather than a physical issue with the leaves. But I don't know.
Yeah. No, that's an excellent answer. And then Mary Huffman has a question regarding a possible management way of addressing slowing the spread of the pathogen, wondering if prescribed fire burning might be a possible treatment.
Well, I think the same concerns that we saw in these wildfires would play a role in the prescribed burning. It would have to be a hot and thorough prescribed fire that was hot enough to really incinerate and consume all infected material, and it would have to be a large enough burn that you would be getting all the area that was infested, because even if you leave a few bay laurels, for example, which are these highly competent sporulating hosts, if you have just a few of these on the landscape, even if the rest of the landscape was very burned, they're harboring the pathogen, they're sporulating, and as all of these plants that are adapted to fire and resprout following fire, as they grow back, right immediately following the fire, then they're vulnerable to being infected again, and so the cycle continues. So it really would take an extremely thorough prescribed burn or a wildfire, extremely hot and thorough, to be effective for any length of time at knocking down the pathogen.
Yeah, and I could imagine repeated prescribed burning could be another thing to explore, repeated treatments. And then Mark Reedy also chimes in on this question, wondering how that might compare to active bay density manipulation. How might compare to active bay density manipulation.
Yeah, certainly that's an issue that a lot of people are recommending, is, thinning those bays removing bays in areas that you want to protect and to reduce inoculum levels. I think I can't really speak to, it's the efficacy of that compared to prescribed burning. I don't know if anyone can speak to that, but it's certainly kind of getting to the same issue of if you can take out these bays that are the supersporulators and really keep their levels low in an area and very open, then you could potentially diminish pathogen risk in that area, disease risk in that area.
All right. Well, and then Mark's got one more question here. Is there info on maximum or average dispersal distances for sudden oak death spores?
Yes, there is.
Yeah.
It's generally said that the spores generally disperse less than 300 feet, but they can disperse up to 3 kilometers as well. It's shown, in strong wind events, and that quite frequently there's a pretty. Mark, I'm going to send you a good graphics for this, but it's generally thought that small-scale dispersal around 300 feet is like the most frequent dispersal distance, but it's certainly seen that it can disperse much farther than that in big storm events, or even much further than that just on even without big storms. So, yeah.
And then Kent writes that in his experience they've found that frequent prescribed burning kills the oak trees, so that may not be a solution after all.
Yeah.
All right, well, seeing no more questions, and feel free to type in any last-minute ones. I just want to give you a big thanks. Maya, I saw Mark typed in his applause in the chat box, so I'll just echo that here. I know we can't hear everyone, but thank you for doing this presentation for us. And yeah, it's definitely a hot-button issue.
Well, thank you for inviting me, and thanks everyone for attending and the great questions. As I said, you can follow up with my email address there, mmbeh@ucdavis.edu, and I can send you more information about any of this research, or if you have other specific questions about sudden oak death, I can round up answers on that stuff.
Thanks, yeah, and I can send that information and those citations that Maya listed and her contact info and a link to our evaluation survey for this webinar and some of those other web pages out in an email right after this. So thanks, everyone.
Okay, that would be great.
Yeah, and thanks, Maya. I'm going to conclude the audio portion of this webinar, so feel free to chat amongst yourselves if you wish, and have a great afternoon, everyone. Take care.
Yeah.